In molecular bio, there's a thing called antigen-antibody interactions. Antigens are large molecules that are usually proteins on the surface of cells, viruses, and other things. They're abbreviated AG. Antibodies are a production of immune system in response to antigens, and the abbreviation for antibodies are AB. So antigens, since they cause antibodies to be released, you want a good ratio of antigen-antibody interactions. So there are two types of interactions, and the first one is particle interaction. That leads to agglutination, which is an interaction with antigen antibodies. The second, if it's soluble, it leads to precipitation reactions. The reaction equation for the interaction is the concentration of Ag plus the concentration of Ab leads to an antigen-antibody complex, or Ag-Ab complex. There are three different possibilities. Actually, there's four different possibilities that you can have. So the first one is you have high concentration of antigen, and you add that in with a low concentration of antibodies. Now when they interact, you get an antigen-antibody complex that is weak. The second type is when you have a low antigen with a high antibody concentration. When they interact, you also get a weak interaction. The third and fourth one is where you have equal concentrations of antigen and antibodies. This is an either high concentration of antigen and high concentration of antibodies, or you can have a low concentration of antigens with a low concentration of antibodies. Both of these produce a reaction or interaction that is strong. So the types of interactions that these two make are ones that could be hydrogen bonds, or they could be ionic bonds, or they could be hydrophobic bonds, but they just can't be covalent. And if you look at the reaction equation, the reaction is symbolized by a double arrow. This means that the reaction is reversible. If it was a covalent reaction, then it can't be reversible, which makes sense. So to visualize the different possibilities that you can have, in the first one you have a high concentration of antigens with a low concentration of antibodies. So antigens are drawn in as red circles in this case, and they're connected together. There's a lot of antigens in this picture, and then antibodies are drawn as orange, and they're kind of like connectors. So when the antibody connects, there's a lot more antigens to antibodies, so that results in a weak interaction. The second possibility is where you have a low antigen concentration with a high antibody concentration. So in this case, let's say there's 3 antigens, and there are more antibodies to the antigens. So all of the antigens are filled up. This results also in a weak interaction. The third and fourth one is an equal interaction between antigen and antibodies. So if you have a high concentration of antigens and a high concentration of antibodies, antigens are drawn in as red, and the antibodies are drawn in as the orange connectors still. Now what results is a strong interaction that's preferred, and this is called a antigen-antibody lattice. That's because they're all connected together and it forms a strong preferred bond. So basically you want to have an equal antigen to antibody ratio. The ratio is a really important part because you don't want to have too many antibodies to antigens or too many antigens to antibodies. You want a perfectly equal concentration, which produces the antigen-antibody lattices. That's the key point of the interaction between the two and what the two are.